In the humid air of West Africa during the 1700s, a Swedish naturalist named Carl Linnaeus pressed a broad, star-shaped leaf into his journal. The castor oil plant grew wild around him, its spiny seed pods hanging like clusters of alien fruit. Local villagers showed him how they pressed the pale beans to extract a thick, clear oil that fueled their lamps and soothed inflamed skin. But their demonstrations carried a sharp warning: they would never crush the raw beans for food. Holding a single, mottled bean between thumb and forefinger, an elder explained that to eat it was to invite a swift and terrible death. Linnaeus watched as they handled the seeds with deliberate care, a respect born of generations witnessing the plant's dual nature. Back in his makeshift study, Linnaeus meticulously documented the dichotomy. He described the useful oil, but his notes emphasized the bean's hidden peril. He recorded the local names and the symptoms they recounted: violent cramping, internal bleeding, collapse. The castor plant, Ricinus communis, was catalogued not just as a resource, but as a vessel for a poison without a name. Nearly a century after those West African warnings, the toxic secret within the castor bean finally surrendered its identity. In a German university laboratory during the 1880s, a chemist named Peter Hermann Stillmar focused his microscope on the bean's pale flesh. He wasn't interested in the oil. He hunted the poison itself, methodically separating proteins from fats and sugars until one compound consistently proved lethal. Stillmar dissolved his purified extract into a clear solution. With a steady hand, he injected a minuscule amount, barely a pinprick's worth, into the flank of a laboratory rabbit. The animal initially seemed unaffected, nibbling at its food. Then, over hours, its breathing shallowed. Its muscles weakened until it could no longer stand, a paralysis creeping through its body that ended in quiet death. In his published papers, Stillmar named the isolated protein ricin, derived from the plant's scientific name, Ricinus communis. His meticulous notes recorded a terrifying potency. A dose smaller than a grain of salt could kill a dog. The world now had a scientific name and a measured lethality for the bean's ancient threat, transforming folk knowledge into a documented chemical weapon. The German scientist's discovery did not remain confined to academic journals for long. By 1917, as trench warfare consumed Europe, the United States War Department took notice. Military researchers in American laboratories began examining ricin's potential on the battlefield. Their objective was chillingly simple: to weaponize a plant toxin against human beings. In one lab, scientists experimented with coating shrapnel fragments in a concentrated ricin solution. The theory was that even a non-fatal wound could become lethal as the toxin seeped into the bloodstream. In another, they tested methods for dusting bullets with the fine, deadly powder. Each test required meticulous handling, with researchers working behind protective glass, measuring the precise dose needed to guarantee a kill. Yet the project soon hit insurmountable obstacles. Ricin proved unstable when exposed to the heat and friction of a fired bullet, often degrading before impact. Delivering it effectively as an aerosol over a battlefield was equally impractical. By the war's end, the program was quietly shelved, the files stamped and stored away. Ricin had failed as a conventional weapon, but the seed of its potential had been planted in the military imagination. The Cold War's shadow stretched into the damp London streets in September 1978. Georgy Markov, a Bulgarian writer living in exile, waited for a bus near Waterloo Bridge. A man bumped into him, then apologized. The tip of his umbrella had pricked Markov's thigh. Markov felt a sharp sting, but the man melted into the crowd, leaving only a small red welt on the dissident's leg. Fever gripped Markov that night. By morning, the wound had swollen into a hard, angry lump. Doctors at the hospital were baffled as his temperature soared and his white blood cell count plummeted. Over 3 agonizing days, his organs began to fail. His kidneys shut down, his heart strained. He slipped into a coma, his body waging a war against an invisible, relentless enemy. An autopsy finally revealed the assassin's tool, a platinum iridium pellet, smaller than a pinhead, drilled with microscopic holes. Inside that metal sphere was a trace of ricin. The pellet, fired from a modified umbrella, had dissolved just enough to release the toxin directly into Markov's bloodstream. The Bulgarian Secret Service had weaponized a laboratory protein into a perfect, deniable instrument of murder. The Bulgarian umbrella assassination had demonstrated ricin's potential as a tool for state-sponsored murder. But in the spring of 2003, authorities in South Carolina confronted a different reality. Inside a dented aluminum trailer, a metalworker named Kenneth Olson had turned his welding skills toward a new project. He wasn't a spy. He was a civilian, working alone, and he had succeeded. The air inside the trailer was thick with the acrid smell of solvents. On a makeshift bench, a coffee grinder sat beside a stack of glossy, mottled castor beans. Olson had followed crude instructions found online, pulverizing the beans and then using a series of filters and chemicals to separate the deadly protein from the plant matter. The result was a small vial containing a fine, off-white powder. When federal agents raided the property, they found the vial, the beans, and his handwritten notes. Olson had produced less than a gram of weaponized ricin in his improvised lab. His arrest sent a shockwave through law enforcement. The knowledge and materials to create a biological poison were no longer confined to state laboratories. They were now accessible in a trailer park. From that South Carolina trailer, the trail of ricin led north to Washington, D.C. In mid-October, a routine day at a Senate mail sorting facility shattered when a clerk slit open a standard envelope. A puff of fine white powder escaped into the air, dusting the sorting table and the clerk's gloved hands. The facility's alarms blared, freezing every worker in place as the first emergency protocols engaged. Hazardous materials teams descended, their sealed suits a stark contrast to the mundane mailroom. They isolated the letter, its postmark now evidence. Portable detectors hummed near the powder, their readings causing technicians to exchange grim nods. The substance matched the signature of ricin. Within hours, the directive came: evacuate and seal the entire Senate office complex. Staff streamed out into the autumn air, leaving behind desks, coats, and a building now treated as a crime scene. Decontamination crews moved in, their process meticulous and slow. They scrubbed every surface the letter might have touched, filtering the air and bagging potential evidence. For days, the heart of American legislative power stood silent and empty, its operations paralyzed not by a bomb or a bullet, but by a few grams of plant-based powder mailed in an envelope. The investigation now had 2 threads: the powder in Washington and the man who had already proven he could make it. The Senate mail facility had just recovered from the scare a decade prior when, in April 2013, another letter triggered alarms. This one, addressed to Senator Roger Wicker, contained a coarse, off-white powder that preliminary tests flagged as ricin. The discovery sent a fresh wave of dread through Capitol security, prompting an immediate lockdown and the familiar grim deployment of hazmat teams to the sorting floor. Federal agents traced the envelope's postmark to Memphis, Tennessee, and then south to the small town of Corinth, Mississippi. There they found their suspect, an Elvis impersonator and local actor named James Everett Dutchka. In his cluttered martial arts studio, investigators discovered castor beans, a coffee grinder coated in suspicious residue, and printing materials that matched the threatening letters. Dutschke's defense was bizarre. He claimed he had mailed the letters as a twisted prank, believing the powder was harmless. Forensic evidence told a different story. The powder was active ricin. A jury later convicted him, not for a joke gone wrong, but for knowingly producing a biological weapon and attempting to use it as an instrument of terror through the US mail. The actor's misguided prank in Mississippi was not the end of the ricin letters. 5 years later, in the autumn of 2018, a different kind of envelope began its journey through the mail. This one was addressed to the Pentagon. Its contents were not a white powder, but a coarse, granular substance that shifted inside the paper like sand. Screening protocols at a government mail facility caught it. A second, nearly identical envelope bound for the White House was intercepted soon after. Hazardous materials teams, their movements deliberate inside protective suits, isolated the letters. Portable detectors hummed, and initial field tests flagged the granules as a biological agent. The samples were rushed to a laboratory for confirmation. The lab analysis identified the toxin as ricin. The FBI traced the letters back to Utah, where investigators arrested a man in his late 30s, a former Navy sailor. In his home, they found castor beans and evidence of crude processing. The motive appeared to be a personal grievance, his attempt to weaponize the mail echoing threats from years past, yet born from a new, isolated resentment. The alarm sounded the moment the letter passed through the sorting machine. Postal workers froze as a supervisor sealed the area, their routine shattered by the threat hidden inside an ordinary envelope. Within minutes, the first hazmat teams arrived, their bulky, sealed suits a stark contrast to the fluorescent-lit mailroom. They moved with deliberate precision, isolating the contaminated section from the rest of the facility. Inside the sealed perimeter, a technician extended a wand connected to a portable detector. The device hummed, its sensors analyzing the fine, off-white powder that had spilled from the torn envelope. A readout flashed, indicating the presence of a biological toxin. This preliminary field test was only the first step. It confirmed enough danger to justify a complete lockdown, but not enough for a definitive identification. The sample, now sealed in a triple-contained vessel, raced under police escort to a high-security laboratory. There, scientists in a biosafety Level 3 suite performed the confirmatory tests. Using advanced immunoassays and mass spectrometry, they pinpointed the protein's signature, ricin. The analysis revealed its concentration and purity, forensic details that would later help trace the powder back to its source. After the decontamination crews had packed away their gear, the legal framework remained. Ricin was classified as a Schedule I controlled substance, placing it alongside weapons-grade biological agents. This meant its possession, outside of a few licensed research facilities, carried severe federal penalties. Yet the very plant that produced it, the castor bean, remained perfectly legal to buy, to grow, even to decorate a garden. This paradox created a persistent blind spot for authorities. A would-be poisoner didn't need a sophisticated lab or rare ingredients. They needed only a handful of beans from a common ornamental plant, an internet connection, and a crude kitchen setup. The toxin's recipe, though dangerous to execute, circulated in shadowy corners online, a dark echo of the scientific papers published over a century before. So the threat never fully receded. Every few years, another envelope would surface in a mail sorting facility, its mundane exterior belying the refined powder within. Each incident reinforced the same unsettling truth. The most significant barrier to ricin wasn't access to the source, but the unpredictable intent of the individual holding the seeds.